Our last speaker is Lou Wei from Caltech, good friend and collaborator. Lou, I think the floor is yours on metabolomic Raman imaging. I don't see Lou coming on.
Moved. Okay. Now.
Oh, there you are.
Oh, okay. Here I am. Okay. So, can you see my screen? Okay. Hopefully, yes. Well, oops. Hi, everyone. My name is Lou. It's my great pleasure to join this webinar and share with you some of our work on spatial metabolic imaging via functional bound selective microscopy. So throughout the central dogma, I would like to argue that the molecular identities becomes increasingly complex, more complex. So we have heard two beautiful talks toward spatial genomics, transcriptomics, with a proper sequence-based amplification that's now achievable, and with antibody-based capture mechanism, spatial proteomics also on its way. But one challenge for doing small metabolites-based imaging toward spatial metabolomics has been really, really challenging to achieve. This is because you don't have really good mechanism to capture different structures of different metabolites, and they're really small. If you think about the way to detect it, you can use fluorescence, but then if you tag the metabolites, it's going to be really perturbative. You can develop fluorescence sensors, but then usually you can only develop one at a time, and that requires the knowledge of the full genome of the system you're working with. You can do, on the other hand, you can do mass spec-based imaging for spatial metabolic imaging. However, that it really suffers from low throughput and low spatial resolution. So one of the promising technology that could contribute significantly towards spatial metabolic imaging is by harnessing the molecular vibrations, so what we call vibrational spectroscopy and imaging. So in this case, you can harness the molecular vibrational motions using either Raman scattering or the infrared absorption to probe different chemical motions that gives you chemical specific information and for probing small metabolites. Especially if you add small vibrational tags that gives you better sensitivity and identity, and with minimum perturbation, we can also probe the metabolic dynamics in this case in live cells. Advantage of doing vibrational-based detection is that all the vibrational peaks are really narrow. So in essence, they are about 100 times narrow compared to fluorescent peaks. So in this case, it shows really high promise toward highly multiplex detection towards spatial metabolomics or other types of omics too. Of course, this technology has its challenge. The traditional issue with spontaneous Raman scattering or mid-infrared-based absorption is the signal is relatively weak. So we usually suffer from low sensitivity at the range of millimolar, and also without the tagging in the typical label-free fashion, we would run into high cellular fingerprint background issue. Specific to infrared absorption, it also has low spatial resolution that doesn't give you subcellular resolutions. So one of the ways we and others have been working on is trying to harness the nonlinear molecular excitation to amplify the signal while make the technology also accessible as an imaging tool. So this get rid of the requirement of other enhancement mechanism requiring extra materials or surfaces. So make it more compatible to intracellular imaging and for live cell imaging. Of course, with matching development of chemical tags that I will show you in a bit, we can add more functional information to retrieve the spatial metabolic dynamics. So in this talk, I will show you a technology called stimulated Raman scattering microscopy, and with this novel coupling with minimum perturbative chemical probes. I want to show you some one work of toward metabolic studies. If I have time, I also want to show you a new type of technology that we recently developed that we call bonfire, that harness the infrared excitation coupled with fluorescence excitation, so that we would be able to map the vibrational signature at the single molecule region toward spatial omics applications. Moving from linear spontaneous Raman to nonlinear stimulated Raman, you need one additional laser for excitation. And that could facilitate the weak Raman transition by about 8 orders of magnitude, really overcoming the low sensitivity issue. This technology was developed actually not 16 years ago now, and it's making its way to be more popular toward metabolic imaging. In terms of setting up the microscope really looks like confocal fluorescence. In this case, we need a relatively special laser that are 2 lasers running to the picosecond. One is a near infrared OPO laser that runs from 700 to 990, and the other is 1030 nanometer. So we route the 2 lasers into the microscope, where you have your molecule of interest, the chemical bond of interest, sitting on top of that, and you match your wavelengths of 2 lasers to match with this vibration transition frequency. So in this case, the specific chemical bond will be excited, and it gives you a what we call stimulated Raman loss signals that can be detected by a large area photodiode. With high frequency modulation and point scanning, you can generate a chemical map through that way. So that's how you can retrieve chemical specific information to differentiate one metabolite to another. So in this case, I'm showing you a Raman spectrum of a cell. You can target a CH3 bond, which is what we typically attribute to the methyl group of proteins, and map out its protein distribution in label-free way. You can also target the CH2, the methylene group, which is usually enriched in lipids, to map out its endogenous lipid distribution. Similarly, phosphodiesterase for DNA, AMID-PIC for another protein band, and rimbrenzin-PIC for phenylalanine. What's more interesting, what we focus on doing is trying to introduce small vibrational tags that are bioorthogonal and highly non-perturbative. So in this case, they are carbon deuterium bonds, which shows in the spectra in a highly specific cell silent region. So if you have used a deuterated amino acids to incubate the cells, and then when cells are synthesizing new proteins, you would be able to capture its protein synthesis activity core space, or over dividing cells in the neuron, or even to identify the spatial hotspots in tissues. In this case, this is a hippocampal tissues in the dendrogenesis. You can also use deuterated glucose and to feed the cells, so that, and then target carbon deuterium vibrational bonds to trace where the carbon deuterium end up with, and using that characteristic peak to assign where the newly synthesized lipids or proteins are being distributed inside the cells. You can use even a triple bond, the carbon-carbon triple bond, as an analog for DNA synthesis, developed analog for lipid tracking in a highly non-perturbative way. So with that overview of stimulated Raman for metabolic imaging, it has really been demonstrated to be a good lifestyle compatible subcellular microscopy way to capture the chemical composition, synthesis, conversion, and transportation and degradation of the metabolites. So with that, we teamed up with Professor Jim Heath, trying to develop Raman into a Raman-guided subcellular pharmacometabolomics tool to understand metastatic metabolic heterogeneity in metastatic melanoma cancer cells. And we're more interested in capturing some of the unknown metabolic susceptibilities. So we use this a panel of human-derived melanoma cancer cells that if you analyze their transcriptomics information, they can be differentiated into 4 different phenotypes: melanocytic, transitory, neurocrust, and undifferentiated. And that also indicates their difference in the trends in their innate or induced drug resistance. However, if you do similar exercise of those metabolic genes, you categorize them into different groups based on their different phenotypes. However, there's no unique metabolic pathways or metabolic categories that stand out. All the metabolic pathways spanning across all different metabolic pathways. So instead of continue that analysis, we resort to Raman spectral imaging. First of all, we mapped out the whole Raman spectrum at the single cell resolution, and we coupled with unsupervised surprisal analysis for dimension reduction. What's interesting is that in this dominating dimension analysis, that we saw a constraint, that we saw a very obvious spectroscopy signature indicating this peak, CH2 peak as a lipid and CH3 peak as a protein. So its lipid protein ratio really decreases significantly from melanocytic phenotype to the undifferentiated phenotype. We further capture this in live cells using serum Raman imaging by taking the ratio images between the lipid channel and the protein channel in a label-free fashion, and we saw this beautiful change decrease in terms of lipid protein ratio toward more undifferentiated or more drug-resistant phenotype. We use this trend as our mapping criteria to map against the transcriptomics, and we saw that a couple of lipid-regulating genes stood up. And that's very consistent with our map, with our data. We also further map it to gene ontology pathways. And one of the pathways that stood out is the fatty acid biosynthetic process. You see this very nice linear trend between our Raman measurement and the gene enrichment analysis. So we hypothesized that across these different phenotypes, the decreasing of lipids may likely due to the de novo synthesis. So we use a live cell assay of a deuterated glucose for incubation, and then we target a carbon deuterium band that specifically corresponds to the newly synthesized lipids once the carbon deuterium is being transferred to newly synthesized lipids through TCA cycles. And we also confirm this decreasing trend that really confirms that the lipid to protein ratio was most likely come from this decreased fatty acid synthesis across the phenotype. We further confirmed that this is the drug is also effective, matching the same trend. So this exercise really confirmed that at a single cell level, Raman and transcriptomics can really have good consistency. But we are really interested more in the subcellular features using Raman since we have a 400 nanometer resolution at the subcellular level. So we wanted to do the surprise analysis at each subcellular pixels, but that turns out to be more challenging. So we decided to take a simpler approach that we performed the analysis at this very bright spots here, and they call lipid droplets. The role of lipid droplets are more being recognized not only as a storage, but more as a harbor as a harbor for lipids, mediating its degradation transportation. So we got the Raman spectrum at each spots across different cell types, and then we perform the same analysis. This time, what the feature stands out very clearly is this unsaturated lipid peak corresponds to carbon hydrogen that is specifically linked to the carbon-carbon double bond. So that's a feature of unsaturated lipid. It also has an increase of cholesterol ester for this undifferentiated drug-resistant phenotype. So the first question we ask is whether the subcellular measurement really corresponds to the bulk level lipid unsaturation in lipids. So we got the pallet of cells and extracted the fatty acid and asterized them, run the GC mass. However, there's no consistency. So if you look at the data, there's no increase on the unsaturation across major lipid species. So it puzzled us for a while. It became more clear to me once we started to do lipidomics. It turns out the cluster ester and triacylglyceride, they do have significant increase in unsaturation for the M381 cells, the unsaturated, the undifferentiated phenotype. And they are the major lipid droplet resident species. However, their total percentage across the whole lipid in cells is only less than 6%. So all the unsaturation signatures got averaged out. If you do it at a single cell level, you have to map this at a subcellular level. So then we use the drug to inhibit the monounsaturation and the polyunsaturation, trying to pin down which one is underlying the susceptibility. Both drugs are effective because you see this decreasing peaks in Raman. However, only by inhibiting the monounsaturation, you saw the cells become lose viability. So this is consistent if you do ssRNA. We further identified using fatty acid analysis that this we are mostly blocking the transition between stearic acid to oleic acid. So we did more of a time lapse of apoptosis. You see this kinetic decay effect that the cells, they don't become apoptotic after after about 48 hours of inhibition. And this is the same as you do time lapse, time-dependent experiments. One of evidence we got that also point out to our suspicion is that this seems to be either something's being accumulated or something's being consumed to contribute to this kinetic delay effect. So what we observed is that if we use the monounsaturation inhibitor, we start to see these very bright signatures showing up at subcellular level. If we use control or using the polyunsaturation inhibitor, they're not there. So one experiment we did is we did the Triton wash, which is known to be extract all the membranes, as the membranes are very fluidic. So on the top, this is the same, and at the bottom, the same cell field of view before and after Triton wash. You know, all the fluid membranes are being extracted while the bright spots are being retained. And by doing in situ Raman spectrum, and specifically this bright areas, we concluded that these are just pure membrane. They're phase-separated membrane structures and excluded all the membrane residing proteins on that structures. So this indication of formation of solid membranes, phase-separated solid membranes, were very excited to us. We also confirmed this through the de novo synthesis pathway. We're adding more of a excessive amount of saturated fatty acids. We further did lipidomics, and it becomes more clear to us that if you look at 6 major species for the lipids, triacylglyceride, if you inhibit their monounsaturation, their saturated fatty acid content actually doesn't change much, but their unsaturation becomes significantly decreased. While all other 5 species, their unsaturated species actually doesn't change much, while the saturated species, saturated fatty acids become significantly increased. So that sort of gives us a general metabolic picture that this very aggressive phenotype of melanoma cancer cells, they, for some reason we don't know yet, they like to store unsaturated fatty acids in the lipid droplets. That's mapped out by our Raman work. And when we start to inhibit their monounsaturation, it's going to induce imbalance of saturated fatty acid and unsaturated fatty acid. And they're going to release unsaturated fatty acid to compensate for that imbalance. That's why we see this decrease here. Once it becomes two terminal, the saturation becomes dominated. They form this saturated solid membrane, phase-separated solid membrane structures. That's what we saw as the very bright spots in at our subcellular Raman measurement. So with that, we further confirmed by adding more unsaturation using oleic acid incubation, we actually were able to rescue the susceptibility for those M381 cells. We further did lipidomics through control cells treated with the monounsaturated inhibitor and rescued by oleic acid. And the pathway that stood out is NF-kappa B pathways that we're currently trying to understand more. But I hope that I have shown you, give you an idea of how powerful those subcellular Raman-based pharmacometabolomics pathways platforms can be used to understand biology in cancers and hopefully in other systems too. If I have 1 more minute, I probably will take this 1 more minute to just give you an idea of what one of the new techniques we're trying to develop. So I've shown you that one of the advantage of Raman is to have this very narrow Raman line width, and you can make them spaced out towards omics-based imaging to break the so-called color barrier. However, this still, we are constantly limited by sensitivity. But you want to work with biology, you usually have to really work at low copy range, at ideally single molecule. So we recently developed a new technique that is really a fluorescence-based technique, but it harnesses the vibrational excitation using infrared excitation. So it's a hybrid technology that we call bond selective microscopy that gives you a chemical bond selectivity by using this mid-infrared excitation and gives you single molecule sensitivity by using fluorescence detection. So I'm going to skip some of the details, but I want to show you that with this, we now can do single molecule detection. Each of the dots you are seeing here is one molecule sitting on top of a film, and we can map out their spectral, vibration spectral corresponds to the chemical bond signatures for this dye molecule. Now we can also do a wide field imaging, but now we can do for the different isotopes that edited on the nitrile bonds. If you do it fluorescence, once you conjugate it fluorescent dyes, if you do fluorescence, they're really non-differentiable. But if you are based on their vibration spectroscopy, you can differentiate their identity at a single molecule level. And here in the field of view is each of dot is single proteins labeled with one dye. So with that, we can do now very bond selective imaging with harnessing the narrow bandwidth, and we can do more on live tissues and then also multiplexing on cells. So with that, hopefully I'm good on time. I want to thank my group and also the funding resources, and I'm happy to discuss more at the Q&A session. Thank you.